Hello everyone and welcome to Blood and Hematology YouTube channel. This video is about morphology of eosinophilic maturation sequence and tissue eosinophil. Eosinophils originate from multipotent hematopoietic stem cells in the bone marrow. Multipotent hematopoietic stem cells give rise to CD34-positive common myeloid progenitors, which differentiate into granulocyte macrophage progenitors. Under the influence of specific transcription factors, granulocyte macrophage progenitors commit to the eosinophil lineage and become eosinophil progenitor cells, identified as CD34-positive and interleukin-5 receptor alpha-positive. The earliest CD34-negative eosinophil progenitors express the eosinophil-specific marker EMR1. During subsequent maturation in the bone marrow, eosinophil precursors undergo a series of morphological changes, becoming promyelocytes, myelocytes, metamyelocytes, and finally bands and segmented mature eosinophils. Using light microscopy and Romanowski staining, the first eosinophil maturation phase that can be identified as eosinophilic is the early myelocyte. The cytoplasm is weakly basophilic and contains a small number of large dark red-violet pro-eosinophilic granules, and numerous specific granules, the orange color of which is fading into the blue cytoplasm. The nucleus is relatively immature, eccentric, and with little condensation of chromatin. Pro-eosinophilic granules stain with a violaceous hue. Mature eosinophil myelocytes are characterized by the presence of large, pale, and reddish-orange secondary granules, along with rare primary granules. The cytoplasm is colorless to pink. The nucleus is usually round to oval and contains a coarse chromatin. The nucleoli usually are not visible. Eosinophilic metamyelocyte has a colorless to cream-colored cytoplasm, with few primary granules and many secondary granules. The nucleus is indented and kidney bean shape. Indentation is less than 50% of the width of the hypothetical round nucleus. The nuclear chromatin is coarse and clumped, and nucleoli are not visible. The eosinophilic band cytoplasm is similar to the metamyelocyte stage. The nucleus is constricted without thread-like filament. Indentation is more than 50% of the width of a hypothetical round nucleus. The mature eosinophil is slightly larger than the neutrophil. The background cytoplasm is colorless and contains characteristic refractile, orange-red secondary granules. The nucleus usually has 2 to 3 lobes. Tissue eosinophils. Smears of bone marrow can contain large cells with a well-defined reticular chromatin pattern, nucleoli, an irregular cytoplasmic outline, and cytoplasmic granules similar to the reddish-orange granules of blood eosinophils. It is thought that these cells are tissue variants of the more motile eosinophils of the peripheral blood. Tissue eosinophils also can be found in tissue other than bone marrow. Thank you for watching this video. If you liked this video, to give it a thumbs up and share it. Do not forget to subscribe to the channel, if you still have not. You can click on the bell to receive notifications whenever a new video is uploaded.